So high throughput screening, this is also abbreviated as a HTS. It is used for the further lead optimization. It will become a input for the lead optimization. So in this, we are going to have the following contents, which contains method of lead identification, combinatorial chemistry, where we are going to analyze a parallel synthesis, and high throughput screening. We analyze all compounds which are having the activity in a mixture we can do. Then we will find out what type of segment which is going to give as a drug-like moiety. And we do the in silico lead discovery techniques without using the lab, that is test tube, or in the pragmatic approach. We are going to do in the in silico, that is in the computers. So the above one combinatorial chemistry and high throughput screening we are going to do in the practical labs. Hit identification will be resulting. Then we are going to convert this into a pharmacophore. This pharmacophore further modified, and QSAR we are going to do. Then we are going to do the molecular docking. In that, we are going to have a energy minimization and conformational analysis, and a conformation of a particular shape we select and go ahead with that particular moiety for the clinical trials. This is all sequence of the events which we are going to have in the computer-aided drug development. So in this particular journey, first we need to start with a lead. So what is lead? A lead is a compound generally defined as a new chemical entity that could potentially develop into a new drug by the optimizing its beneficial effects and minimizing its side effects. So this is a lead compound, is not a drug that you need to consider. It is a new chemical entity, and it is not named as a now, it is still chemical entity only. Whenever we are calling it as a chemical, not as a drug, we don't have any idea of the toxicity, and we don't have any idea of its beneficial effects also. It means efficacy and toxicity, we don't have much guarantee on functioning. But that can be developed into a new drug. This NC will become a new drug by optimizing the beneficial effects and minimizing the side effect. Then it will become a drug. So the drug, what we are going to have is a new drug because it is already not existing. That is the reason we call it as a new drug. Or we are making a drug a modification which is giving some benefits which is better than the existing drug. So once a target, a testing system has been chosen, the next to find the lead compound which shows the desired pharmaceutical activity, where we are going to have a beneficial effects dominating than the side effects. Then we will contain the after one another. Pharmaceutical activity. The first step in this particular journey is lead identification. So the identification of the small modulators, molecular modulators of the protein function, the process of transferring these into high content lead series are the key activities in the modern drug discovery. So directly, we cannot build the scaffold of a drug directly. We need to take a protein function, and the protein function we want to modulate it. Either you want to shut down or we want to shut on. So these proteins are responsible for so many diseases, their malfunction. We want to manipulate them and modulate them. So this process we are going to do in the reversible manner, so that our disease will be cured without having a toxic side effects. And in this particular case, we are going to choose the small molecules. So these one will be transformed to a series of compounds, and we are going to have their activities so that we can find a better option to select for that treatment of a disease. So a lead compound is first foothold on the drug discovery ladder. So first, whatever we are going to put is that one, it is a lead, it is a starting of our journey. So then in the next 1,000 miles and millions of dollars and multiple years it will take to come out of a drug. Because of the intervention of the computer-aided drug design, we can reduce the cost also, we can bring down the timeline also drastically, because we are going to find the failures in advance. It takes much more effort to take the lead compound into a drug candidate. It is not an easy journey. We need to have a multifunctional team where we are going to have the computer specialist, pharmacists, chemistry persons, and also math component will also be present. Statisticians are there, and we are going to have the artificial intelligence interventions, machine learning algorithms. So all together, we are going to have a journey smooth to get a new drug into the market. How to find a lead compound? So what are the possible routes? Because our journey is to get a lead, that is a first fold in the climbing of a drug discovery ladder. So first step, we want to take it where to put that first step. That is the one which you need to change. There are 7 types we can take so that our journey will be guaranteed to come out of a drug, so that we will not have a pitfalls and up and downs. So the one possible, there are the different possible routes in the directions when we are moving it, we can identify the lead with guarantee. So one is the chance observations. So this is just like a serendipity. We want to see something, but we have found something else. It is better, and which is working, and it is promising. So this chance, we want to take it as a choice, and we will go ahead. The second route is selective optimization of the side activities. Selective optimization of the side activity means we are having already one existing drug that is not working because of their side effects. So these side effects, we find that there is a promising for the treatment of another disease, and we optimize further to get a drug for that particular disease. So it is actually a planned activity only, not like a serendipity. The SOSA approach is we want to take the side effect as a main stream, that is main path, and modify it to come out of a drug. So the sildenafil for the example, which has come like this only. So because the sildenafil, which is used for the treatment of the cardiac diseases, so it is turned into a different moiety, and all of a sudden it has come into a different market approach, which has come because of its side effect. So this is used to treat the erectile dysfunction, and the story is big, and $1 million business has come. So this is actually what the selective optimization of side activities. Another one is herbal and folk remedies. These are all used age-old phenomena from our grandfathers and forefathers and ancestors. They were having some treatment methodology which is lying in the home itself and nearby places itself. So these remedies are working very well for the treatment of the disease, but they don't know what is the mechanism of action, what are the principal constituents which is responsible for the treatment of disease. So we use those folk medicines only as a starting points and extract them, take a water soluble extract and lipid soluble extracts, and we take fragments and we are isolate the main components. Then we will come out of a particular active constituent, that main ingredient which is responsible for the treatment of the disease, and we will come out of the parallel composition which is also responsible for some another remedies and some another purposes. So we are going to isolate them after extraction, isolate them and screen them and find out the disease treatment abilities in that particular mixture. This is the journey which is open in multiple areas and doors. So each route what we are suggesting, it will open the many doors. So we can enter into any door so that we will be getting drug into the market with success. That is a guarantee. The fourth route is screening of natural product metabolites. So most of the times the herbal drug what we are extracting, that constituents what we are taking out, in the screening we find that nothing is working. We get confused. Why this is all components we have screened, but it is not working. But as a remedy, we are able to get a success there. The reason is we are screening the actual one that may be a prodrug, but after the metabolism, the a prodrug converted into a drug that is acting. So now our journey is going to take a analysis and review and screening of the product metabolites. They are responsible for the treatment of disease. So the screening of natural product metabolites, now it will work with guarantee instead of wasting our time from the extraction and separation techniques and screening techniques. These are all not going to work because they are all the prodrugs. So the metabolites, when we are taking them, that will give the idea to modify further so that we will have a better potency and less side effects, and we can have a more efficacy. Another way is the fifth route is the screening of compound libraries. We have got say around 10 to the power of 90 compound are there, 10 to the power of 60 libraries are present, and 10 to the power of 20 libraries got evaluated. So these are all the journeys keeping on going. So we have got so many chemical compound libraries, and we take those compounds and we do the high throughput screening in the laboratory. So by have a parallel synthesis approach, and we take them and we screen them and find any chances of having any activity in the treatment of the disease. Any disease, we will use them for the screening for multiple diseases, and we will find out later how to climb in one of the step what we have taken as a one door we are going to enter. Another approach is called as the rational drug design. So in the rational drug design, we have got a ligand based drug design. Another one is structure based design. In the ligand based design, we don't know the structure of protein pocket. We don't know, but we know the ligand which is already working. We want to modify it further, bringing a better moiety into the drug by the bioisosterism and having a similarity index. And we have got a science backup for whatever we do it. And rationally, we go with the ligand based design. And in that, we can have a fragment based drug design also. The next one, which we are having a structure based design. In this particular case, we are having a new disease, and the protein responsible for the disease is known, but we don't have any drug like COVID-19. So we know the disease, but we don't have drug. In that structure based design are very good. So we take the protein structure and fit a ligand suitable for the particular moiety. That is particular pocket. We are going to do a science based approach for the getting a success. The last one is a seventh route, that is the natural substrate based design. So we are all failing all the 6 routes. We are not able to approach, but our body has got a defense mechanisms and immunity, and they have got intrinsic chemicals. They are responsible for the combating the known diseases. Like we are having some of the scaffolds and the series of enzymatic reactions for the apoptosis. So we are having so many gene mutations, correction of the gene mutated genes. So we have got so many inbuilt hormones and enzymes. So they are working as a substrate to the structure of proteins, and they are making a altered change according to the environmental responses and disease states and immunity, antigen based immunity. So these are all going to help us. So what we do now, we take them as a track, and the track will lead to a door. When we open the door and entering into the big world of drug invention, that is new drug invention based on the natural substrate. So if you choose any one of this in the design and development of a new drug, you are going to have a success. Without having these approaches, what will happen? You are hitting in the dark. So when you hit in the dark, arrow will fall on what object we don't know. That is the reason we need to focus on a particular path so that we can guarantee to reach that particular goal. So we need to take these 7 types of the routes to identify the lead compound. Now we will go further. Our journey now, it is present in this particular presentation, is the 5th route, that is screening of compound libraries, that is called as a HTS, high throughput screening. So this is going to start in the lab, not in the computer. HTS is not going to work in the computer. First, we are going to start with a lab where without some reliable measure of a potency, it is impossible to conduct the systematic analysis of a possible new drug. Reason is, even though we are going to have certain computer simulations and artificial intelligence approaches, and we have got some possible routes, but there is no guarantee that they are going to work in the real time in the human being. That is the reason we are going to start with reality and we are going to expand it for the betterment. So that is the reason we are going to do the screening or assaying after making a compounds in the lab. Really, we are going to do them and in the pragmatic approach and take them for the screening for the multiple diseases. And wherever the compound which is having a parallel synthesis, which group, functional group and series of functionals groups which are going to act, we will select them and we will go for that. So the testing of series of molecule is very much important. We are not going to, in the high throughput screening, we are calling it as high throughput. We want to see this type of functional scaffold, whether it is going to work as a drug, we want to study and remove it because we want to have fast. So there are so many compounds, millions of compounds are existing in the libraries, and we can't make all of them also. That is the reason we decide to have a certain functional group branches, and these series of compounds we will do, and we are going to screen them against the biological target that correlates with the cellular pharmacological activity. This process is called as a screening. Why we are calling it as a screening? We don't know before in what disease this chemical is working. We don't know that. That we are going to find out. So later onwards, we will find the mechanism of action, like whether it is working with the enzyme inhibition or it is receptor binding. And this is all going to come once it is working. Then we go with the mechanism of action of it is working. So this is the one journey makes it as a guarantee that our success is going to lead a fruit. Otherwise, the entire effort is going to futile. So this is now made possible easier in the modern research because macromolecular targets like proteins, enzymes, receptors that can be genetic receptors, which is based in the DNA and RNA, they can be identified with ease. Targets are now available in the large quantities using in the molecular biology. So we can screen them so that we can come out of a drug. So the automated high throughput screening technologies are also available. So that is the reason we are going to have these compounds synthesized with robotics. And we are going to synthesize a series of compounds. We know the molecular biological cell lines and what the tissue cultures, we know them. So we are in the hand. So series of chemicals we have and a series of cell lines and cultures, tissue cultures we have got. In laboratory itself, we can find their functionality and we decide whether this route is going to work or we need to take a U-turn to go to another scaffold or the series of compounds. This is actually the fundamental approach and the principle in the high throughput screening. Why you are calling it as high throughput? You are going to have a exponentially very fast and it is completely automated. So when you are taking a well plates and the robotic arms will take the sampling by their own and it will be making a synthetic routes and it will be giving the result when the next day it will give the report on the printer. So you can see what are the functionality without your intervention. That is the reason it is automated, completely automated. High throughput screening technologies are now available. So what are the approaches will finalize and what we are going to have in our sequence with our high throughput screening, we will have a linkages now. So one is combinatorial chemistry. And we are going to have, it is not a straightforward general chemistry or a specific chemistry. It is a combinatorial chemistry where we are going to get a series of compound at one shot. That is called as a combinatorial chemistry. Don't think that I'm going to evaluate a single compound with guarantee. It is not required now. We want to have a combinations of synthetic molecules that is going to be available in one shot. Then we are going to screen them. That is the reason we are calling it as a high throughput screening. All these series of compounds were given to the multiple targets, multiple screening approaches. That is also automatically. The third, next, once you are getting these series of compound, one series has got response in one type of tissue cultures, or we are going to have a screening technique, cell lines. Then we take that particular receptor and see the mechanism of action. And now you take as in silico approaches to make it very fast, because we don't want to go further, and we are going to go with the computer usage with machine learning and AI tools. That is in silico lead discovery techniques. After that particular, when you are going to have, we have got hit identification and go further screening and refining the activity. So the first job in this particular journey is combinatorial chemistry. So what is combinatorial chemistry? Combinatorial chemistry is a technique by which a large number of different types of structurally similar molecules, structurally similar, that you need to understand. So you have got a structure activity relationships, and you have got a quantitative structure activity relationships we are getting to help us to refine our efficacy and toxicity. We are already having that journey known. Now we are going to have large number of different structurally similar moieties. It will be having a scaffold. Main lead is same, but their functional groups are getting varied with respect to your amine groups, amides, and halogens. So they are synthesized by the high throughput, that is combinatorial chemistry, then subjected to the high throughput screening. So this technique uses the same reaction conditions with the same reaction vessels to produce a large number of analogs. So we are going to have with this particular setup, and you may get the question now in your mind, when we have got a mixtures of a series of compounds which are having a similar molecule, structurally similar molecule, we have got a different scaffolds, different series. Then how to select from that? Which series is going to work as a drug? Yes, in this particular journey, why you have got a high throughput in this particular case? So you are going to give the mixture of compound, not an isolation compound or a pure compound to test it. If you are isolating it and purifying them, it will take a lot of generations to come out of a drug. So you give as a mixture. When it is hitting it, then you find which is going to responsible for that hit. That is the journey which will be refined. Number of arrows which are hitting in the dark will be removed, and which is hitting onto the target, we will be taking them only, and they are called as a hit because they are hitting with guarantee. So it means we are guarantee success is present in our journey. Otherwise, $1 million investment, multiple years, we are doing so many man hours we are doing. If this hit is not having, then what is use? So we are going to take U-turn and go back to R&D without going to enter into the market. So this approach is having a guarantee. That is the reason we are going to know this one also as an approach. Now we are going to see the workflow or a flow diagram, which is having in the combinatorial chemistry within the design. So most of the times what will happen, we are going to have a multiple series of the compound which is having a similar functional group, and we are going to have a high throughput screening. We take the lead. After having the lead, then we will optimize the lead for the betterment of the activity, and after optimization, you will come out of a new drug that is developed, which is not already existing. So now this chemical, we are naming it as a drug now. Previously, we are calling it as a hit, lead, pharmacophore. These are all things which will be named as a drug. So reason is, in our journey, entire journey is based on increasing the efficacy and decreasing the toxicity. So when you are balancing it, when the toxicity is under the control, and efficacy is going to reach high potencies, then it is chances of having and revolutionizing in the market as a drug is more, and getting the returns very fast. So this entire approach, which will guarantee us without any failures, and the failures also we will find in early stages. Now, what are the synthetic methodologies for this production of the libraries of a structurally similar compounds? Synthetic methodologies we need to learn, otherwise how it will be possible. So nowadays we are having a green chemistry because we want to have environmental friendly techniques, because standard sustainability goals we have got. So that is as an impetus which is being revolutionized the entire world. We need to see our benefits linked to our globe impact also. So the first technique in this particular case is a solid phase techniques. So most of the times the environmental impact will be present with the solvents, what we are going to use for the reaction mixtures and washings, and we are separating them and recycling them. It is taking a lot of impact onto the environment. So the solvent may be a toxic, or it may be a ozone layer impact, or it may be having a global warming impact, and or we don't have any isolation or treatment techniques that is going to cause a environmental pollution with respect to air, solid, liquid, that is a ponds and rivers, and the flora and fauna of the environment. That is the reason solid phase techniques are the promising, which is called as a green chemistry, where we use a less number of solvents, and their quantity is also in the minimum. So this is one approach you need to keep it in the mind. The second one is parallel synthesis. In the parallel synthesis, so you have got a Houghton T-bag procedure is present. So the T-bag procedure and automated parallel synthesis and automated parallel synthesis of the all tripeptides from the 3 amino acids, which are going to have a linkages, we can see, and mixed combinatorial synthesis also we can see, and solution phase synthesis is the last attempt we need to do. So in the solution phase synthesis, we are not able to get a success because Arrhenius theory says that most of the products are formed when the reactants are moving and they hit with each other, collide with each other. But there is chances of the collision in the solid phase is very less, because they are in the solid phase, they are confined to a particular particle. So you want to have a reaction progressing when they are having a collision with each other. So you need to use a solvent. Otherwise, the solution will not be formed and collision will not be taking place. And we are able to see that this is not getting a synthetic route, the solid phase, we are not getting a product. It means that we need to use some solvent there. It means it is pushing us to use that. In those cases, as a last option, we need to use a solution phase synthesis. So the first one, which is very much important, and everybody in the world is looking at to convert all the solution phase approaches to solid phase techniques, because this is going to reduce our bioburden. At the same time, it is going to revenues are going to be generated by reducing the solvent usage. So the solid phase techniques includes the reactants are bound to the polymeric surfaces. So they are, we can call it as immobilized and modified when still they are attached to the immobilized support. The final product is released at the end of the synthesis. So this we are removing it. The same immobilized polymeric surface used for the further synthesis. So this is what the concept is. So that we are going to have the similar vessel we are going to use for the multiple recipes. Like that we can use it instead of purchasing disposable one, or we are using multiple vessels. So it is going to take a effort for the further investments. So here what we use the subtract is a resin bead. So the resin bead are the functionalized surface, it acts as a solid surface. So where it is going to anchor, or it will form as a linker. The bond linking to the substrate is a linker, where we are going to have our catalysts or our required chemicals which are linked there. The bond must be stable in this particular case. It should not be detached. Otherwise, we cannot make a immobilization. If it is not immobilized, we are not able to do it. We need to make it again and again, like a regular synthesis, it will become. So the bond must be stable to reaction conditions used in the synthesis. It is preferably covalent bond, not like a electrostatic bonds or hydrogen bonds or van der Waals forces or London forces. So we need to have a covalent bonds. A means of cleaving a product from the linker at the end. This is also very much important. If you are not able to remove the product from the substrate, it means it is got dead. So we are not able to remove the product. That is also required to see, so that at the end of the reaction, we should be able to remove our, wash out our product. So protecting the groups of the functional groups are not involved in this particular synthesis, because we are protecting the groups by the linking, and we are going to have a multiple scaffolds of the similar types of synthesis. We are doing it. Similar parallel synthesis we are doing. So the procedure is beads must be able to swell in the solvent used and remain stable. And most reactions under this bead interior only, and which is going to result in a product formation. Later onwards, we will extract or remove that particular product from the resin or the bead. You see, in this particular picture, you are making it a resin which is appearing like a ball, and we are going to allow it to swell in the solvent. Then it is swollen. It is having a porous and swollen nature. It will open the multiple pathways, and the starting materials and the reagents and all are going to get linked there. Then we are going to have a product formed, and product is delinked later, and we are going to get the washing. Further purification will be done if necessary. So what the advantage is that specific reactants can be bound to the specific beads. So the same beads will be used for the multiple reactions and synthesis that is required in the high throughput screening. So beads can be mixed and reacted in the same vessel also to will get a multiple mixtures of the compounds. And products formed are distinct for each bead, and physically they are distinct, and we can separate them by based on the size, based on the color coding or the dye, we can separate them. Excess reagent can be used to drive the reaction to the completion, and excess reagent and the byproducts are easily removed because we can filter them, because the bead is naturally a macro in size. Reaction intermediates are attached to the bead and do not need to be isolated and purified. Only the product what we are isolating is the product one only. Accordingly, we are going to have a washing solvents. Individual beads can be separated to isolate the individual products, and each bead can be separated later, and we can separated beads can be, again we are going to use a specific wash solvent to get the separate products. So the polymer support can be regenerated and reused for the cleaving of the, after cleaving of a product. We can use these beads again, put it into the new flask, and again we do the resynthesis. Like this, we are going to repeat the same substrate for the multiple products, and we are going to yield a multiple savings by using the similar one by the regeneration and reuse. This is what the concept in the solid phase synthesis. Now we will aim at the parallel synthesis. Just we will go back and see our attendance. Good, 26 persons are there. I will take the screenshot. Now coming back to our session. So if anybody is having a doubt, they can put it in the chat box also, and they can unmute themselves and they can ask in between. So as usual. So coming back to our parallel synthesis. So the parallel synthesis, the aim is to use the standard synthetic route to produce the range of analogs with the different analog in each reaction vessel, tube or the well. So most of the times it can be a tube where you can have a continuous synthesis pathway, It can be a vessel. It will provide the little bit larger quantities if you want to synthesize, and you can optimize the volume also. And sometimes it is a well, a small microliter volumes are present, so that your revenues are because your consumption of the chemical is very micro quantities. Reason is you are not synthesizing for the production. You want to synthesize for the research and development. So initial stages you are going to have in the microgram quantities only. So by this well method, the advantage is that your reaction vessel become multi-liter vessel to microliter wells. That is the reason it will have a multiple synthesis routes. When you are doing it, it will consume very less quantity of the chemicals. So the identity of the each structure is known because we are using the similar moieties in the way each well. So we can know the structure what we are having in each cell. Useful for producing the large number of analogs for the structure activity relationships and other optimization. Say for example, when we are taking the analogs where we are taking the insertion of the amino groups, whether the activity in the high throughput screening in the cell lines, whether they are decreasing or increasing, if you are able to find out, or there is no change in the activity. So then it will act as a SAR, structure activity relationship. Bringing the amide group or the amine group or sulfur group or hydroxyl group or carboxylic group in the analogs. So where we are going to have a change in the activity, whether it is a positive or a negative, or it can be no change. So we are going to find out that in the high throughput screening, and we can assign each compounds which are present analogs, whether they are having a negative impact or positive impact or no impact in the activity. So this is where we are going to have a parallel synthesis success. The entire analogs, it will give the idea that in that particular disease target, this is going to have what type of impact. So we are going to study accordingly. We will choose them for the further studies. So in this T-bag approach, Houston T-bag approach procedure, each T-bag contains the beads. So the beads which are having a certain reagents which are present. So the reagents are already embedded there. And we are going to take, and each bead is labeled with respect to their size and the dye or the color code. They are labeled to have a multiple compounds which are going to be yielded as a product. So the separate reactions are carried out in the each bag, and combined T bags for the common reactions and working procedures, and we can use them. And separately also we can, that in one T bag we can use the multiple regions also. A single product is synthesized within each bag, then they need to, then there is no need of a confusion. If you don't have a similar analogs, then you can have a single product in each bag. So different products are formed in the different bags. So they are taken for the screening later. And which bag is going to work in a particular target, and after screening we know it, and we will proceed what the reactants which are present in that bag. So we can go ahead so that we are going to use a similar approaches multiple times, and we will get a treatment options for the multiple targets and the diseases. That is the reason it is having an economy in its functionality and workflow also. And if you want to have in one shot, you can have a combined T bags. So the combined T bags, say for example, you have got a 10 types of the beads in one T bag. So the combining of a T bags, what will happen if you don't have any target hitting with respect to this particular all 10? So this we will leave it aside. So next 10 box is having some positive impact in the target. So first 10 are gone now. The second 10, which may be any one of that may be having, or two of that we having, we don't know in the combined T bag approach. So the economy of effort is there when you don't have the activity, positive activity. In one shot, the negative activity can be raised. But the T bag approach, when you're doing in one way, it is good. But the reason is in this individual T bags, each activity of each T bag, what you have got a product will be known. So that we can find out. But all 10 you can't do one in single tea bag approach. That is a bead which is present in the tea bag. Then what will happen? 10 times you need to report, all 10 will get a negatives. So in combining effort, in one shot, you will be removing all 10. That is the advantage. And the disadvantage is that even one is present. How to identify that? Again, you need to do the individual tea bags where you are having a activity. This is the one which is one. Maybe a 10th tea bag, maybe is individual when you are doing it, may be a functional. Then even if it is a sixth one, if it is a functional, you go further because it may be another tea bag also containing some products, which is also maybe having in the activity in the similar receptor. We can have a different scaffold. It means all 10 we need to do to confirm that whether the activity is coming from all or not. So in the combined tea bag, even though it reduces the number of efforts, but once it is getting a positive result, we need to assign what the positive result, where it is coming from. So even though it is like this, it is very cheap and possible in any lab because of we are using a beads. If you want to make it bead on your own way or purchasing from the standard laboratories, we can use for the particular scaffold synthetic routes. Manual procedure is not suitable for producing the large quantity of different products. Singly, we are doing like a trial and error method, and randomly we are picking up and doing. It takes a lot of time. That should be avoided in the high throughput screening. Automated parallel synthesis. In this case, we are going to use a synthesizer which are containing well plates. So reaction vessels, there can be a 42 or 96 or 144. So they are having like a completely as a set. And in that we use the beads and are the pins which are used for the solid phase supports, depending upon the shape what we are taking. Reactions and the workups, we can do it combined way or individually what you are using like a tea bag. And these well plates, automatically they will be taken by the synthetic robotic arms. They will be going for the further screening and evaluation for the screening techniques. So the same synthetic create used for the vessel, which will be used in the water. Generally what we are going to use, we can alter in the wells different reagent conditions also to optimize the better synthetic platforms. So different products are obtained each well per vessel. We consider that like a vessel. So each well, then we are going to take them for the further evaluation to see that the feasibility of a synthetic pathway and feasibility of the screening to have a drug-like moiety also can be assessed at this particular stage itself. So we will proceed with the third method, that is a VAST, that is a combinatorial chemistry tomorrow. We will wind up the class now. If anybody is having a doubts, they can unmute themselves and they can ask.